So in the last section, we talked about Mendels Laws. And in this section, we're going to talk about variations on those laws. So sometimes alleles don't show complete dominance over one another. And you can have incomplete dominance, which is the appearance of a hybrid that's somewhere between the two parent phenotypes. So if we have a red flower here with that's RR, it's true breeding, and we have a white flower that's little r, also true breeding. The gametes given off by this red flower are going to be all capital R. The gametes given off by the white flower are all going to be little r. And when they combine, they're going to make this pink hybrid. And the pink hybrid is capital R, little r. And it will have half of its gametes will be capital R, and 1/2 of the gametes will be small r. So if we do a table, we will get the sperm here, and there'll be 1/2 big R, 1/2 little r, and the eggs over here, 1/2 big R, 1/2 little r. So we'll get a red plant with red flowers. We'll get 2 that'll have big R, little r, and will be pink, and we'll get 1/4 that will be white with 2 small r's. Sometimes, instead of being dominant over each other, genes can be codominant. So the A, B, O group, blood group phenotypes in humans are controlled by 3 alleles, which give us 4 phenotypes. So we have the IA and the IG alleles are codominant. Both alleles are expressed in a homozygous individual. So if you have an IA and an IB, you'll have type AB blood. So let's look at how this works. If you have blood group, it's up here. So if you have phenotype A, your genotype is IA, or it might be IA, and the carbohydrate produced is this triangle, which is carbohydrate. We'll just call it carbohydrate A, carb A. If you have blood group B, your genotype is either IB, or it might be IB, and then an I, which doesn't have any carbohydrate associated with it. And you'll make these round carbohydrates. So this is carb B. If you're heterozygous, you have blood group AB, and your genotype is IA and IB, and you make both carb A and carb B on your blood cells. If your blood group O, then you have just II, there's no A, no B, and you have no special carbohydrates added. Okay, so sometimes a single gene can affect many phenotypical characteristics. This is Pleiotropy, and the example given in your textbook is sickle cell. Sickle cell causes damage to organs. It causes pain, fever, joint problems, anemia. The advantage is that heterozygous persons are resistant to malaria. And so 1 in 10 African Americans is a carrier for sickle cell because it has this benefit to people who have just one copy of the gene. And sickle cell causes this is a normal cell, normal cells to sickle, and they get, they don't flow through the vessels properly, they get stuck, and you have all sorts of trouble. You can have polygenic inheritance is kind of just the reverse of pleiotropy. In polygenic inheritance, a single characteristic is influenced by many genes. In pleiotropy, one gene influences many characteristics, so they're complementary to each other. But they're also non-Mendelian, where in Mendels processes, one gene led to one characteristic. When we talk about the characteristics, we need to remember that environment is very important to the phenotype expressed. Here we have a family grouping, so they're all related to each other, and they have varying skin tones based on sun exposure. And I like this picture. It says the couple of the daughters of the author, and they're identical twins, but through the use of hair dye and clothing choices, they look quite different.